Before getting into the cancer hallmarks, let's make it clear how a normal cell transforms into a cancer cell. It first undergoes hyperplasia, then dysplasia to become cancerous. In hyperplasia, there is an increase in the number of cells that looks normal under a microscope, while in dysplasia, there is an increase, but they look abnormal under a microscope. So the cancer cell hallmarks includes replicative immortality, inducing angiogenesis, resisting cell death, sustained proliferative signaling, invasion and metastasis, evading growth suppression, genome instability and mutation, tumor promoting inflammation, deregulating cellular energetic, and evading immune destruction. So, what is replicative immortality? Cancer cells typically exhibit increased telomerase activity, which helps them to become immortal, that is, to divide indefinitely as long as proper nutrients and oxygen requirements have been met. Normal cells have normal telomere lengths and divide for a finite numbers of replication. Now, what is angiogenesis? How it is induced? Angiogenesis is the formation of new blood vessels. Tumor cells overexpress vascular endothelial growth factor, which is a major angiogenic factor responsible for the creation of new blood vessels that provides nutrients to the interior of the tumor. These blood vessels are architecturally different from the normal blood vessels, being less organized. So, how the cell resists death? The p53 tumor suppressor protein is encoded by the p53 gene in humans. In normal cells, p53 suppresses abnormal growth in part through interaction with pro-apoptotic protein BAX, whereas in tumor cell, mutation in the TP53 gene lead to loss of tumor suppression, loss of interaction with BAX, and inhibition of apoptosis. Apoptosis is a programmed cell death, serves as a mechanism to prevent the accumulation of mutation or damage within the normal cell. Cancers sustained proliferative signaling by expressing activating mutation in proteins involved in cell growth. For example, 50% of melanomas have mutation in gene coding for serine threonine kinase BRAF, and of these mutations, about 90% are V600E point mutation. Now let's see how invasion and metastasis work. Tumor cells spread from the original site of the growth to the distal site through the circulatory system. This movement of the cancer cells from the original site is termed metastasis. Tumor cells are transported via blood or the lymphatic vessel to colonize the distant organs. In some cases, metastasis occurs when tumor cells spread in the body cavities, such as peritoneal cavity, or when tumor cells are inadvertently seeded during surgical removal of a tumor. Metastasis is added by deregulation of epithelial adhesion molecules such as E-cadherin. Now, how cancer cells evade growth suppression? It is done by altering the tumor suppressing signaling protein like p53, which is often mutated, that leads to increased proliferation and growth. Genetic instability consisting of loss of heterozygosity, chromosomal rearrangements, and more, that favors continual growth of the tumor cells. For example, mutation in genes like TP53 are recessive. Thus, loss of heterozygosity leads to expression of mutated p53 protein sufficiently to sustain growth. How inflammation helps tumor grow? The immune cells that promote inflammation infiltrate the tumors and help them to grow by providing growth, survival, and angiogenic factors. And finally, alteration in cellular energetics. Cancer cells require increase energy consumption to fuel their growth. For this, they mostly use glycolysis instead of mitochondrial oxidative phosphorylation. This upregulation of glycolysis is due to increased expression of HIF1 and HIF2 protein under the hypoxic condition. In summary, the cancer hallmarks include replicative immortality because of the increased telomerase activity, induction of angiogenesis because of the overexpression of vascular endothelial growth factor, resisting cell death and evading growth suppressor due to the mutation in the tumor suppressor protein like p53, sustained proliferative signaling as a result of activating mutation in protein like BRAF, invasion and metastasis through blood and lymphatic vessel to the distant organs through the deregulation of epithelial adhesion molecule like E-cadherin, genome instability consisting of loss of heterozygosity, chromosomal rearrangement that favor continual growth of the cancer cell, and the last, tumor promoting inflammation, where the immune cells provides growth, survival, and angiogenic factors. Thank you so much. Keep liking and watching.